Okay, hi everyone. Thanks for coming out to my presentation. So the project I worked on this summer is the bionutrients project, and like Joanne explained, the hurdle that astronauts face is long-term missions is nutrient degradation in food over time. So to deal with this issue, we developed a way to produce nutrients right there in space, and I'm going to explain how we did that and what my part was. So this is some background on the topic. The objective was to develop a platform technology with single-use hydratable packets that contain two ingredients. One would be the food microbe engineered to produce the target nutrient, and the other is the edible growth medium that would be rehydrated and allow the microbe to grow. More specifically, we looked at the production of two carotenoids, lutein and zeaxanthin, and these are usually found in carrots and green leafy vegetables. And the microbe that we looked at was the yeast, Saccharomyces cerevisiae, which is regular baker's yeast. So why did we choose carotenoids? Because they are important nutrients for the eyes, and they protect against diseases like cataracts and macular degeneration. And the reason that we chose these specific nutrients is that astronauts tend to have a lot of problems with their vision after long-term space missions. And why did we choose a strain of Saccharomyces cerevisiae that is spore-forming? Because spores have long lifetimes, they're resistant to temperature and radiation extremes, which makes them suitable for use in our project. And is this project important? Yes, because of the degradation of regular nutrients in long-term space missions, so we had to come up with a creative way to produce nutrients in space. And is it relevant? Yes, this is a recent news article talking about how the vision risk to astronauts could be a potential showstopper for the upcoming Mars mission. So my task in the project was to design and test a disposable bioreactor for the production of nutrients in space. So this is just a picture of what a typical bioreactor looks like. As you can see, it's very large and bulky, probably heavy, and not practical to take up in a space shuttle. So what we worked on are small packets that are disposable, so we can control the quantity of product being produced each time. It's easier to dispose of a packet than worrying about cleaning a large bioreactor like this. And the concept of operations is that the packets will be stored in a cool, dark place on the shuttle. They'll be rehydrated when they're ready to be used, shaken briefly, and then placed in an oscillating incubator to allow the spores to germinate and grow, producing the nutrients that we want. Then the astronauts will consume the contents and dispose of the packet. So this is just a very simplified pathway, a very simplified diagram of the biosynthesis pathway in plants and bacteria, showing the main compounds that we're interested in. So you have lycopene going to alpha carotene and beta carotene, which then goes to lutein and zeaxanthin, respectively. And the reaction stoichiometry is relatively simple. Beta carotene and alpha carotene have the same chemical formula because they are just isomers, and they're going to lutein and zeaxanthin with the help of the coenzyme NADH. So another reaction that that takes place in the bag is the fermentation of glucose. And the main product that we're interested in for this reaction is the carbon dioxide, because of course if you have carbon dioxide being produced in a bag, it will expand and there's a risk of explosion. So that's something that we have to pay attention to. I wanted to ask a question of the audience just to keep people engaged. Can anybody tell me the name and chemical formula of the alcohol produced during fermentation? And if you get it right, I have some space ice cream as a prize from the NASA gift shop. So, anybody? The name and chemical formula of the alcohol produced during fermentation. Mm-hmm. Yes, that's correct. So I'll give you a space ice cream afterwards. Okay, so in light of all this, some of the research questions I had to think about in designing and finding a packet was what would be the best way to oxygenate the bag contents? How would the contents be rehydrated? What should the size of the bag be? How would gas exchange be facilitated in a microgravity environment where liquids and gases don't separate normally like they do here on Earth? And how would the dry media be sterilized? And during the first 2 weeks of my internship, I did a lot of literature review, just looking at papers online with research on Saccharomyces yeast, online research into potential bag designs with the stipulation that any bag that we use had to be produced within the US, so it can be from China, all those great places. And then we had a lot of group discussions, just bouncing ideas off each other. And then finally, I found something that I presented to the group, and everybody thought it would possibly work. So I contacted the company, and we found this packet with a pressure lock system. My mentor and I, we had a teleconference with the VP of Marketing and Sales at the company, and of course they were thrilled because who doesn't want NASA to use their product for a research project? So the pressure lock system, it's a flexible pouch with a controlled dispensing technology. So what that means is that the user just squeezes the pouch and the fluid goes into a separate dispensing chamber, and then the packet automatically reseals, which means that you can have the packet lying on its side or upside down without unintentional dispensing, which is perfect for a microgravity environment where things are constantly moving around in the bag. So after a couple days, they sent us some sample packets to run experiments on, which is really exciting. And the first experiment I did was just to test the bag and make sure it did what it was supposed to do, which is vent the carbon dioxide. So I set up an experiment with a regular Tetra Pak bag with a cork and the pressure lock bag, and I incubated some of our cell culture overnight. And the next day, I don't know if you can see it, but the Tetra Pak bag actually exploded from the pressure buildup, and the pressure lock system remained intact, which meant it did what it was supposed to do, and it worked, which was great. And then I made some alterations to the bag design using a heat steel machine that we had in the lab. So I removed the gusset at the base to reduce the volume, and then I made a slant at the top in order to direct the carbon dioxide out of the bag more easily. But another hurdle that we encountered was that when the bag expanded with carbon dioxide, a kink formed at the top that inhibited gas escape. So what I did was try to make a rudimentary side support to keep the kink from forming, but again, when the pressure built up, it unstuck and the kink formed again. So we contacted another lab downstairs, and Dr. Hogan was able to make an aluminum side support that was custom made for the bag, and it actually worked pretty well. So these design experiments just showed us that having the bubble dispenser at the side was a problem, so we can upgrade the design by moving it to the center and probably heat sealing it upwards. So another aspect of things surrounding the bag was media sterilization, because of course everything has to be food grade in the bag and you don't want microbial contamination. So the media is composed of 3 powders. It's YPD media, so it's yeast extract, peptone powder, and dextrose or glucose. And what I did was make a very concentrated solution of the media and then try to dry it to a powder similar to the way honey is dried. The first method I used was lyophilization. This is a lyophilizer. And the way it works is it removes water from a frozen solution by the process of sublimation. So you put your frozen solution into the lyophilizer, and the water, the ice, converts to water vapor, and it leaves behind your frozen product. And as you can see, we did not get a dried product at the end of the process. It was more like a honey-like mixture, which was not suitable for our purposes. The next method I tried was leaving it in an oven for a few days to see if that drying method would work in various containers. Again, it produced a honey-like substance. Then I tried a vacuum oven, which is just an oven with a vacuum attached to speed up the drying process. And it was a little better at drying it than the regular oven, but still not exactly what we wanted. And before I gave up, I tried a speed vacuum, which is a machine that uses centrifugal force, a vacuum, and heat to speed up the process of evaporation. And as you can see, that is not a powder. So what we learned from this was that none of those methods are suitable, and this is all a part of research. You have ideas and you try it, because you never know until you try whether or not it will work. So another path that they will pursue is looking at a spray dryer, which is a method used to produce powdered honey, and if all else fails, radiation. Another aspect of the bag design is sizing, because if you have bags that are really big, it's not very practical to take a bunch of those up in a spacecraft. Also, if you have 1 liter bags, we can't ask the astronauts to drink a liter of our little potion every day. So the things that we know about the bag is that we want 10 milligrams of lutein per day, 2 milligrams of zeaxanthin per day, and the compounds in the bag would be the dry media, the dried spores, water, and we wanted 50% headspace to allow for carbon dioxide production. And then the unknowns are the lycopene production rate and conversion efficiency, and then we don't know the amount of the desired nutrients initially present in our spores, because that would affect how much we wanted to make. So since there were so many unknowns, and even from looking at research papers, there was like a wide range of possible yields. So it was difficult to pinpoint an exact size for the bag. So instead, what my mentor asked me to do was develop a template to calculate the bag size. So it might look simple, but this took a long time, and it has a lot of calculations involved in it. So all they need to do now is fill in the values for the unknowns when they get them, and they'll be able to determine the bag size. Another theory that I wanted to test was whether adding a compound called ergosterol to the bags could be done instead of shaking the bag, because we read in a couple of papers that people had done that before. So of course you can't just take everything you see in papers, you have to try it out. So ergosterol is a component of yeast and fungal cells, the cell membrane, and what it does is protect the membrane integrity, so there's no need for a cell wall, and the cells can move around and change shape. And these are results from the experiments. So I compare the cells growing with ergosterol and no shaking, growing with shaking and no ergosterol, and then a control experiment. And all the experiments showed that shaking produced better growth than adding ergosterol. And the conclusion from this was just that aeration or oxygenation is more important than adding the ergosterol. And one of the main impacts of shaking the bag is oxygenation, other than redistributing the contents, because when you shake, you kind of oxygenate different parts of the solution. So the next question was, how would we aerate the bag? How do we oxygenate our bag's contents? So we thought of a fish tank type of aeration system, basically having a straw inserted into the bag and connected to an external oxygen source, and that would bubble gas into the contents during germination and provide oxygen to the organism. There's also chemical reactions that produce oxygen, but generally superoxides and chlorates tend to be explosive, so safety issues would invalidate this option. And then there's the option of membrane oxygenators, where you have a hydrophobic microporous membrane with a gas and a liquid side, and somehow that could be incorporated into the bag design. So the last set of experiments I did before I left Ames, because they had a proposal this month that they needed data for, was comparing how well our spores grow to how well vegetative cells grow. So the spores are inactive and dormant cells, and vegetative cells are active. So we just want to make sure that when we rejuvenate the spores, they grow as well as vegetative cells. So experiments were checking the optical density every 2 hours for growth curves, checking the change in dry mass, and also we compared the growth of spores and vegetative cells stored in water. One of the persons in the lab, I have a picture later on, but I worked closely with her for the first couple of weeks, and then she went off on vacation, and then my mentor went off to a conference, so I was kind of working on my own. But it was really good in terms of me being able to work to work at my own pace and plan experiments so that I can attend lectures outside of the lab once I had results to present at our group meetings. So We were working as a team, bouncing ideas off each other.
With the first experiment that you did, right?
Mm-hmm.
With the one that you did with the carrots, that one, right? What conclusion did you come to? Or did you come to any conclusions where you believe that it was possible that it might have a positive effect on the astronauts on the site? Did you come to any conclusion like that?
I'm not sure which experiment you're talking about because my experiments were on the bag design and making sure that conditions are suitable for our yeast to grow. There was a geneticist working on the genetic engineering aspect of it, but because they were trying to meet milestones for the end of the summer, everybody sort of had their separate assignment to work on. So in terms of whether they produced the vitamins or what level of efficiency they achieved as yet, I can't really speak on that.
How did you just completed your
Undergrad, yeah.
How did that research experience compare to your typical undergrad your lab
Right.
experience
Well, at NYU, I worked in the chemical engineering lab, and the research I did there was protein engineering research, so that was more genetic engineering type work. And then this project was more focused on the hardware aspect of the project and, running experiments and doing an iterative process with designing the bag and so forth. So it was a different part of research, of the whole research project and planning a research proposal and all that. So it was different, but I learned a lot from it because that's all part of research.
Sure. Will you be continuing this type of research at the PhD level? Do you see yourself, has this sparked a longer term interest in this kind of research?
Well, the research area that I'm interested in specifically is renewable energy technologies. So I was looking into the area of biofuels and genetically engineering organisms that can convert algae and things like that into fuels. So I would still stay in the biotechnology field, and this kind of gave me a view of another side of research, like I said. So I'm still very much interested in research. Is anybody going to give me a prize for all the questions I'm answering?